vitro biology that they can bring to bear on the game. The other thing I'll say is that companies like ourselves are not doing off-the-shelf assays. We're doing custom work, so we can set the assays up ourselves with or without guidance from you. And we're not doing a lot of this. We're not doing any of this really for the NINDS program, so I'm going to not spend any more time on that. I've already alluded to the importance of the computational part of the exercise here, not only in hit generation, but also in the future hit to lead and lead optimization phases. And so you have all these tools that are available, and these can be contracted. Then the in vitro ADMET PK services, you want to get an early read on absorption. Will your molecules cross the cell membrane, first of all? Will they get through the blood-brain barrier? So there's some basic things that you can do here, and we've all talked about the different types of assays, and you can see we have quite a wide array of things that are available to you to use. Also getting a good idea of metabolic stability. An initial run through a human liver microsome can tell you straight off, are you going to have serious metabolic stability issues with your compounds? You like to know that early. Again, it's not necessarily a point that's going to kill your program at this point, but you want to know so you can address it in medicinal chemistry. And then there's the medicinal chemistry, of course, near and dear to my heart. And here, these are some of the things that we do in the hit to lead and also lead optimization stages. So again, this is the computational part of the exercise here is really very important in both of these stages. We do analog design, synthesis, SAR elucidation of, with your, the primary drug target, but also we're optimizing the drug properties along the way as well. At some point in more advanced stages of hit to lead or lead optimization, we're really trying to obtain PK/PD profiles and trying to optimize those and to optimize the drug safety margin as well. And also very important to anything that you're going to do with this down the line is to establish intellectual property position. So this really is going to start to take shape in the hit to lead part of the work. But then in lead optimization, you really have to have the ability to file patent applications to protect your compounds and your IP. And then, of course, it's once you get into lead optimization and your program really has legs to stand on and it looks like it's got a good shot to go, you have to think more seriously about the chemistry. Is the chemistry amenable to scaling up, to being able to supply adequate quantities for your advanced biological testing, for your IND-enabling studies somewhere down the line? So it's not too early to think about that. I'm not going to get into the chemical development because, quite honestly, we're not involved. We do all this at AMRI, but at least within the context of the NINDS programs right now, we're not near that. So this gives you an idea in terms of resource estimates for doing the hit generation here. And our experience tells us that if we're starting from the in vitro biology stage, we're having to set up the assay, the high throughput assay, and to validate that. Then it's about a 9 to 12 month exercise. The first 6 months really establishing the biology. They can go anywhere from 3 to 6 months, and quite honestly, it's 6 months, even a little bit longer than that has been the norm. But then once you've done that and you've run your high throughput screen, and now you're getting into the hit identification, hit validation, hit selection. That'll take anywhere from 3 to 6 months, depending on the number of hits that you've identified to go forward. And Marcy's already done a really good job describing the fee-for-service models, she described the FTE model. Also, you can do some things fixed fee, but there the task has to be very well defined. The experimentals are well known, you know how to synthesize the compound, and the deliverable is very well defined as well. That means, because with fixed fee, when we're doing that, we're doing it at our risk, which means that if we don't deliver the chemical compound or the biological test report or whatever, then we don't get paid. Whereas with the FTE, that's more of a research-based model. Everything has to be developed. And there are deliverables, certainly, with an FTE model, but you get paid all along the way. And then, of course, there's time and materials, which is somewhere in between that, where the risk again reverts back to the customer. In that we'll give best faith efforts to put the time in to do the work, and then the customer will also pay for the materials. There's no guarantee of a deliverable in that case. So I've already mentioned the fact that AMRI and many other CROs can work in an iterated drug discovery manner. And this is a typical iterative drug discovery cycle for lead generation and lead optimization. And you can see the different scientific disciplines that are involved here at the top. This is missing a few things if you're thinking in terms of lead optimization, though, because they're the in vivo components of the work also need to be slotted in there as well. So just saying a little bit more about lead generation, the goal of the hit-to-lead process. So hit-to-lead and lead generation, those terms are used interchangeably. And so here we're trying to identify a lead chemical series that meets a target product profile. Now the expression, the target product profile, has already been discussed considerably. This is probably a little bit lighter in terms of a profile than you're thinking in terms of when you're getting ready to go into the clinic. But all the things that go into that profile will ultimately address what needs to be put into your profile for a clinical candidate. And so demonstrating reproducible concentration response curve in primary assays, both biochemical and cellular, is absolutely critical. That your molecules work by desired mechanism of action, and that can mean in this case, if you're working towards a specific receptor, is it an agonist? Is it an antagonist? Is it whatever? And also in terms of enzymes, the type of enzyme it is, allosteric, whether it's an active site inhibitor, reversible, irreversible, etc. So also you want to have confidence that your molecules have selectivity over closely related counter targets. You're working in targets that are part of receptor or enzyme families, and you want to make sure that you're hitting the ones that you want. This is especially relevant in kinase type of targets. You want molecules to be devoid of undesirable structural features that are going to cause headaches down the road, either in terms of reactivity or metabolism or things like that. And very importantly, you want to build in the drug-like properties into your molecule that are going to make it readily absorbed and get to the site of action where it has to go. And the biological activity should be responsible to structural changes at the multiple sites in the molecule. This is really what the medicinal chemist is looking for and wants to get an attractable SAR for the drug target and the drug properties as well. And of course, in this lead generation, you want to really start to show very strong potential for development of intellectual property space. And also, you may run a few early ADMET types or PK type studies to see if the compounds are getting into the animals. So this resource estimate is expanded out beyond hit generation and shows the amount of time and effort that it's going to take. So with lead generation and lead optimization, you can see that they'll go anywhere. Lead generation will be 6 to 12 months, and lead optimization 12 to 18 months. And you can see that the amount of FTE support that you're going to need for this goes up substantially as well. So intellectual property, I've already said a little bit about this, but as CRO, one of the primary responsibilities that we have is to develop IP for the customer. And so we're very good at doing this. We have an excellent track record. Over the past 10 years or so, we've been inventors on about more than 90 patents, US patents that have been issued. Just because we're also listed as inventors on the patent, the IP belongs completely to the customer by contract, so there's no confusion there. And one of the most important things that we do is to maintain the secrecy and the confidentiality in working with each and every one of our customers. So lead optimization, I'm going to skip over this slide. This gets into, certainly the barriers are higher here as you get further along, and there are a lot of things that you have to do. The only thing I will say about this is you get into having to address more difficult questions. So you really need to demonstrate efficacy in animal models, and showing target engagement, biomarkers or surrogate markers for efficacy is very important here at this stage. And being able to select a compound that's going to be a preclinical development candidate. And at this stage, you really have to have a good read on what your patent application is going to look like. Now, I've got a couple slides here on moving from in vitro to in vivo, and what we're trying to achieve here. Now, we have an in-house pharmacologist by the name of Nick Moore, whom spent about 20 years of his career at Eli Lilly, very good pharmacologist, and has done a lot of the CNS type models that people have talked about today. These are the types of questions that he believes you have to be able to answer in selecting an appropriate in vivo type model, and also determining who to do the outsourcing of the in vivo studies with. And it's very interesting here in terms of in vivo efficacy. He believes that you're better off in this case finding a niche provider who has therapeutic experience in the area that you're interested in, rather than going with a large house that maybe lacks that therapeutic experience. But then when going to toxicology and safety studies, you might be better served to go with larger GLP accredited tox safety CROs. I'm not going to spend a lot of time on that. So you can see that you're trying to fill the gaps in your drug discovery capabilities, and I've described a number of these and really haven't mentioned anything, for example, like the pharmaceutics area, where you might be thinking about what is the solid form of the drug that you're going to develop. Is it going to be a free base? Is it going to be an acid? Is it, what issues do you have in terms of crystallinity, polymorphs? What about formulation aspects? Those are all other things that are way down the line that you'll have to consider. So in terms of the cost of outsourcing, historically, cost has been a significant barrier to the direct use of CRO services by academic PIs. It can be quite expensive to go out and do this. A lot, as we've heard over the past couple of days, there are a lot of avenues open to you now to be able to do that. And with the 80 different academic drug discovery centers or so that are out there, they've got technology platforms that are now in place to be able to answer a lot of the questions that I've put before you today. But also, very importantly, the nonprofits, the foundations, and government agencies like the NINDS and the NIH are making translation of academic innovation to drug therapies possible by providing the financial means for the universities to do this. And this is just we seem to be getting into academic research quite heavily these days. This is an announcement that we made of a preferred provider agreement with Biopontus Alliance. And again, this is to help fund and support academic research. So in closing, why outsource? I think the answers are pretty evident. These are some of the important ones. I think it really allows you as academic investigators to stay focused on what you're really good at, innovation at the early stages. By outsourcing, you're then able to plug the gaps in your drug discovery capabilities. You're able to access technology without going out and spending a lot of money on maybe an LC, mass spec. You're able to then leverage also industry drug discovery experience that comes not only with the NIH, but also with the CROs like AMRI. And you're able to get to proof of concept a lot more rapidly. Time is very important in drug discovery, and you want to take your idea, your program forward as rapidly as possible. And then finally, it'll improve chances of success. And then just finally, I'd like to thank you all for your attention. And if there's anything that AMRI can do for you and helping you achieve your goals, please feel free to contact us. Thank you.
Perhaps time for one quick question. Thank So is your advice, if I understand, for individual academic investigators to access CRO services through an academic center, through the NIH, or to sort of steer away from direct engagement and direct?
Certainly, cost is the huge consideration and the driver here, and I certainly understand that. People were trying to get research done on drug discovery grants that maybe be $250,000 or less, and when you start talking about the cost of working with a big CRO, that can be cost prohibitive to be able to do that with the amount of resource that you need. I'm certainly not trying to turn business away, and there are now ways which we can work more effectively with academic groups, but there are a lot of avenues, there are a lot of possibilities out there. There are a lot of CROs out there, big and small. But the one thing that you're going to want to do is to be able to invest your money, however much it is, into getting the answers that you need, and being able to get the products that you need to move your work forward. So quality, trust, communication, these are all very important things that you have to look for and consider in who you're going to work with.
Sometimes we often hear concerns that when academic investigators come to a CRO directly, they don't always get the same attention or quality of service, maybe I shouldn't say that, as when a big customer comes along. So what is your...
Well, I can only tell you our personal experience, and I know I've been involved writing a lot of proposals for academic groups, only to be spurned at the end with them going somewhere else or deciding to take another tack. We treat every customer importantly, so it's certainly worth calling AMRI and asking the question, putting together an RFP, and we'll respond to it in a very rapid manner.
Thank you very much. So in the next quick 10 minutes, I will try to highlight the features and the opportunities for advancing preclinical therapy development for Alzheimer's disease through the NIA. Just a brief reminder of the enormity of the public health crisis that Alzheimer's is, with currently up to 5 million people suffering from Alzheimer's in the US alone, a number which is predicted to triple by 2050. An additional 5 million estimated to suffer from MCI, and approximately 10% of these will progress to AD each year. So on top of this, as we all know, the therapeutic needs of patients with Alzheimer's disease remain unmet. So to live up to its mission and to respond in response to this urgent unmet therapeutic need, the National Institute on Aging, over the past 7 years, put in place a number of funding opportunities to support drug discovery and preclinical drug development for Alzheimer's disease. The idea being that our goal is to seed a multitude of projects to increase the number of investigational new drugs that can then be clinically developed either by the clinical trials programs and infrastructure at the NIH or picked up by industry. And this is sort of our funding opportunity pipeline. Really, I think we're ideally positioned. We sit on the wealth of basic research discoveries. And so we try to populate this drug discovery and preclinical development trajectory with a variety of funding opportunities. Once an IND is developed, there is chances are, the hopes are, that industry will become interested, although they're increasingly divesting from early clinical development, so that is the new funding gap. But there are a number of opportunities at the NIA for clinical development, whether through the Alzheimer's Disease Cooperative Study, through a pilot clinical trials program, or through the General Investigator-Initiated R01 funding mechanism. Now, these funding initiatives that were put in place were given special attention. They were funded through discretionary funds from NIA directors set aside. We put special review panels in place, so the required expertise is there to meet the best applications. And it is because of these two things that in the last 5 years we were able to build a fairly sizable and diverse portfolio against a variety of targets. What is currently available focused on AD therapy development, there's 4 major funding venues. The first one is, let's see, these are the exploratory small grants using the R21 mechanism. We have 2 flavors of them. These are for projects that are 2 years long, total direct costs $275K. And here you can jump in at any of these stages of drug discovery, whether it's assay development, providing preclinical proof of concept, doing some initial screening, or doing some initial target validation followed up with some screening. We also support, and this is a joint funding opportunity with a number of other neurological institutes, an R01, through an R01 mechanism, programs that are 3 to 5 years in length. You have the flexibility again to step in here at any stage of the discovery process. You can go some ways into preclinical drug development depending on cost. And so this is a program that is widely used by investigators. Then we have the preclinical drug development funding opportunity that uses a cooperative agreement mechanism. It's 3 to 5 years, and the costs allowed are between $300K to $1 million direct costs per year. And if you are, so here you are stepping in at the stage of optimized hits, or you are selecting a preclinical lead, optimizing it all the way through an IND submission. If you happen to have to be at a late preclinical stage of development, you can take advantage of IND enabling services provided through a contract. Now, I mentioned that these applications in response to these funding opportunities go to special review panels. The R21s and R01 applications are served by Drug Discovery for the Nervous System Review Panel at CSR, and the scientific review officer is Dr. Mary Custer. The U01 applications are reviewed at NIA's review branch, Drug Development, entitled Drug Development, and the scientific review officer is Dr. Alexander Prasadanian. If you have a request for access to IND enabling services, these requests are reviewed by NIA program staff and 2 external advisors with relevant expertise. Just a couple more words about the U01 preclinical drug development program. This is the 3rd reissue of this funding opportunity. The entry bar is somewhat higher than for the R01. Funding venue, here you need to have be at a stage of identifying a preclinical lead. You need to have proof of principle of efficacy in a relevant animal model, preferably against a defined therapeutic target. The program supports a variety, all the potpourri of lead optimization activities, all the way through support for pre-IND, meeting with the FDA, and IND enabling toxicology and safety pharmacology. What is not supported and is not responsive, considered not responsive to this program, is basic mechanistic research, hypothesis-driven applications, or early stage drug discovery activities such as screening. It's a milestone-driven program, so the research plan must include quantitative milestones with specific criteria of no-go, or go and no-go decision-making. The milestones are not only subject of special review criterion, but they are also used by NIA program staff to assess determined progress on the project and to recommend further funding. The services for IND enabling studies are provided. NIA has a contract with SRI International, and the contract provides some of these services, not all. As you can see, analytical, bioanalytical chemistry, pharmacokinetics and bioavailability, preliminary toxicity screen, assistance with preparing for a pre-IND meeting with the FDA, and IND-directed toxicology including safety pharmacology, 14 all the way to 90 days. What is not provided are the more expensive items such as GMP synthesis, CMC, long-term IND-directed toxicology. To apply for these contract services, you need to contact the project officer, which is Neil Buchholz, the NIA, and provide supporting material and data to justify background information on your compound and justifying the request for contract services. Your request is then sent out to the external advisors, and if it is approved, the project officer will put you in touch with the principal investigator at SRI, Karen Steinmetz. SRI will then prepare a protocol based on the information provided by the academic or biotech investigator. Once this protocol is approved by the NIH contracting office, then SRI can proceed and carry out the agreed upon studies. Sort of the time between submission of request and approval of the protocol for these services is between 4 to 6 months. And it really, this depends on what time of the year you're applying. We operate on fiscal year cycles, and that may shorten or lengthen the process. Now, how to decide which funding opportunity is best for you? I do recommend, yes, no one reads the funding guidelines, so I left that bullet out. So here is my advice. Start by at least read who is the project officer, program officer on those funding opportunities, and send us a brief one-pager summary of your prospective application, outline the goals and deliverables, and that would be sent to yours truly. What we'll do then, we'll set up a follow-up teleconference to discuss what is the best funding venue. Hopefully there is a best funding venue that we have at hand. We may, if appropriate, refer you to another institute, such as the colleagues at NNDS, or refer you to a trans-NIH funding opportunity, or to a non-federal funding agency. We'll basically try to find the shoe that best fits. If the project is mature enough to enter the U01 program, then we'll request preliminary milestones and budget, and we'll work with you to develop and formulate the quantitative milestones for the application. And Lorenzo Ruffalo at the Division of Neuroscience works very closely with investigators on these pre-submission activities. If the project is at late preclinical development stage, then our advice will be to submit a request for contract services to Neil Buchholz. So things to remember, I said no one reads, but I cannot emphasize this enough. Please read the funding opportunity guidelines. And these are these inane 10 to 20 page documents, but this is what you need to read. Submission deadlines on the front page. Purpose of the funding opportunity also on the first page. Scope and objectives and budget. And this will save you a lot of time, and it will help us start a conversation on the important topics and and move you along faster. All the useful information of what is responsive and not responsive is in this. This is one page of text. So I really highly recommend that you do that. Also, consult with program staff early and often in advance of submission. We recommend 6 to 8 weeks before an R21 or R01 submission, 10 to 12 weeks before a U01 submission, not only because this is a more elaborate project that requires more back and forth between program staff and the investigator, but also because the price tag on the U01s is often $500 and over, and applications with budget $500 and over undergo administrative review, which needs to be in place and approved 6 weeks before submission. So please help us to help you and follow these sort of timelines if you can. Something that I didn't put in here, but things to remember, is to think ahead and plan both for failure and success, and build your team early. Even if it is an R21 application, you do medicinal chemistry, don't just write that you'll follow the Lipinski rule of 5. Find a medicinal chemist and really give a message to the review committee that you're serious about this effort and that you know what you're doing and talking about. Now, we collaborate with the Alzheimer's Drug Discovery Foundation on a number of projects. A small but meaningful way of collaborating is reflected in this announcement that the ADDF has on their website, and that is that they will consider funding or providing financial assistance, not fully funding, relevant NIA and NINDS grant applications, beginning with applications that have been reviewed in calendar year 2011. These are applications that have undergone review, have been scored, not supported by the NIH institutes, but fall within the ADDF funding priorities. And Diana Shinneman would be the contact person for that. So please take advantage of this opportunity. We are not allowed to send your application elsewhere for funding, but we can certainly, that is another place where you can turn to get just-in-time seed money to get your project off the ground. And that sometimes makes all the difference. One of the important parts of the NIH AD Translational Research Program is education and outreach. And what we provide in terms of education and outreach is not nearly enough, but I think it makes a meaningful difference. This conference is in part supported by the NIA, I mentioned, with co-sponsorship from NINDS and Office of Rare Disease Research. The conference is not only educational for the academic investigators branching into or stepping into drug discovery, it's quite educational for those of us who are directing these translational programs at the NIH. Another educational and outreach venue is the biannual Translational Research Investigators meeting that we hold. We have had 3 of these to date. The investigators that are funded through the U1 program are bound by the terms and conditions of their award to attend these meetings. We still hope they're useful to them, because what we try to provide there, in addition to bringing their colleagues funded through the other mechanisms, is we bring drug discovery experts from the academic realm, industry, FDA colleagues, clinicians, and it's really trying to build a networking hub. This is a network of investigators that can interact with each other and synergize their research programs. It's a chance for us to see how the program is doing, and it's a chance for the investigators to showcase their projects and get very valuable feedback from our external advisors. The program is 7 years old. It is still a work in progress. There are funding gaps remaining. The funding that is in place certainly is not nearly sufficient to meet all the needs. So we're working on a number of things, trying to improve it, refine it, and expand it. We do this with a group of very dedicated external advisors, some of them in this room. I should mention Barry Greenberg, Marty Watterson, Barbara Sahagan, who I think is not here at the moment. So they donate their time very generously, and always we appreciate their candid advice and feedback. We of course appreciate the feedback of our investigators, because we're here to serve them so that we can together serve the public. And these are the NIA translationals, the 4 of us. On the bottom, in addition to me, Lyra Follow and Neil Buchholz, Lori Ryan, who directs the clinical trials program, and we try to seamlessly connect basic research discoveries and feed them into that a clinical endpoint. And so this is the team of 4 that works on the program. Thank you. We'll continue with Rebecca's presentation and take questions as a duet at the end, if there are any.
Thank you, Susanna. I think you've given an excellent overview of the Alzheimer's programs at NIH that support the preclinical stages of drug discovery. There are dozens of other programs at NIH that support research in this stage of the drug discovery and development pipeline. And it's the end of a long day. I certainly don't have time to cover all of those. But I want to highlight a few different examples that I think are going to be of particular interest for those of you doing research in neurodegeneration, and it can give you a flavor of the different types of ways that NIH supports research. I also want to give you some tips for how you might find the programs that are best suited for your particular needs. One of the messages I want you to take home from this is that there are many different ways that NIH supports preclinical drug discovery. There are many different types of funding mechanisms that we use. And we also provide services in kind, so you can get free access to drug discovery services, similar to the kinds of services you could get through a CRO. I'm also going to be spending a few minutes talking about a relatively new and rather unusual program at NIH, which essentially gives you your own biotech company to do all the work needed to advance a project into first-in-human studies. So here I have a few examples of our most popular programs at the Neurology Institute, NINDS, within this preclinical drug discovery space. The ART-21 exploratory grants and the series of cooperative agreement grants are very similar to what Susanna described at the Aging Institute, and these will cover research for any diseases that fall within the NINDS mission, so any neurological disease. We also have 3-year R01 grants that support some of the earlier stages of drug discovery, including assay development and screening, and early stages of chemistry to optimize a compound that you might use